Hi everyone, today we're going to be solving AQA GCSE Biology Foundation Tier Paper 1F. In this particular video, we're going to be solving June 2021. Today we're going to be solving the entire question paper. The first question is about cells. Which diagram shows oxygen moving by diffusion? We can see the direction of movement and we can see oxygen molecule being outside. Now we know that always oxygen is in higher concentration outside the cell and lower concentration inside the cell. So the first diagram is correct, because we can see higher concentration of oxygen outside and lower concentration of oxygen inside the cell. Cells use oxygen for respiration. So any oxygen that enters the cell uses it to burn it with glucose and produce carbon dioxide and water. So the cell runs out of oxygen. Alright. Diagram 2 is wrong because it shows oxygen moving out to a higher concentration, which is definitely incorrect. And in the third diagram, we can see the cell is filled with oxygen, whereas more oxygen is coming from outside, which wouldn't be possible because it is a diffusion gradient, and diffusion is the movement of substances from a region of higher concentration to a region of lower concentration. Complete the sentences. We need to choose the answers from within this particular box. Plant cells absorb substances from the soil. Plant cells use osmosis to absorb. Now, osmosis is a process that is only applicable for water. No other substances we can apply to osmosis, so it will only be water. Plant cells use active transport to absorb. Now you see, active transport will be used on those materials, those that are higher in concentration inside the plant but lower in concentration in the soil water. So in this case, mineral ions will be the answer because mineral ions are lower in concentration outside the plant. However, plant still needs to absorb it, so the plant uses active transport to absorb those mineral ions. Active transport moves substances against the concentration gradient and needs, obviously, active transport, since it does the work like uphill. Active transport is like going uphill, and if we were to go uphill, we would need energy. So active transport requires a lot of energy from respiration. Figure 1 shows a specialized cell that absorbs substances from the soil. In this diagram, we can look into the specialized cell, which is shown. It is the root hair cell. So the root hair cell has a extended protrusion, you can see over here, which increases the surface area to volume ratio by a whole lot, Alright, name the type of specialized cell. So it will be root hair cell. Describe how the cell in figure 1 is adapted to increase the absorption of substances from the soil. The root hair cell has very large surface area because it has a long projection. A sperm cell is another specialized cell. Figure 2 shows a sperm cell. We can see the nucleus and we can see the long tail that extends after the nucleus. Draw one line from each feature to show how the feature helps the sperm cell carry out its function. It contains a nucleus. Alright, so the features are to break the outer layer of the egg, to help the cell to swim to egg, to provide chromosomes for fertilization. We know that nucleus contains chromosomes, so it will provide the chromosome that is needed for fertilization. The sperm cell has a long tail. Now obviously the long tail is going to help the sperm cell to swim to the egg. To release energy, the sperm cell requires mitochondria, and to break the outer layer of the egg, the sperm cell requires the tip of the sperm cell, which contains an enzyme called acrosome. Figure 3 shows another specialized cell. We can see a motor neuron. Name the type of cell in figure 3. Describe one feature of the cell that helps it to carry out its function. Now specifically, motor neuron name is not necessary. We can also call it a nerve cell, and the specialized feature that we can see within itself is we can see that it contains a very long protrusion. Alright, so it has a very long which we will call long axon. Okay, alright, it has very long axon, so it can carry information for a pretty long distance. And we can see there are branches, meaning that it can connect many neurons as well. So the name of the cell will be nerve cell. We can also write motor neuron, by the way. And feature of the cell is that it has branches, or it has insulation, or it is long. Viruses cause disease. What name is given to microorganisms that cause diseases? So, microorganisms that cause diseases are called pathogens. Predators are organisms that kills other organisms, and prokaryotes are bacteria. So, those two are not correct responses. How do viruses cause the symptoms of disease? Viruses engulf white blood cell, destroying them. Viruses are very small compared to a white blood cell, so definitely they do not engulf it. Viruses produce antibodies that damage tissues. Our white blood cells are responsible for producing antibodies, and thereby, this is also a wrong statement. Viruses reproduce inside cells, damaging them. This is correct. The only way a virus can make a copy of itself is by reproducing inside a host cell. So, in that particular process, it takes over the mechanism of that particular host cell and ends up killing the host cell in the process. Figure 4 shows a virus and an animal cell. We can see the protein coat, we can see genetic material within that particular protein coat, and we can see animal cell. In the animal cell, we have cell membrane, we have cytoplasm, we have nucleus. Now, the question says, suggest one reason why viruses are not classed as cells. This is a very good question, and this is a very common question as well. We have to understand that in this particular answer, we see many of the feature and that are present, basic feature that are present in an animal cell are not at all present in a virus. For example, an animal cell contains a cell membrane. A virus does not contain a cell membrane. An animal cell contains cytoplasm. Viruses don't have cytoplasm. Animal cells contain a nucleus, whereas the virus doesn't contain a nucleus. Animal cells have mitochondria, alright, they have ribosomes, whereas viruses don't even have mitochondria, doesn't have ribosome, doesn't do any metabolic reaction on its own. That's why we class it as a it is not classed as a cell, by the way. Okay, so what answer you're gonna write for this particular option? Since there are many options to write, you can write they do not have a cell membrane, and that will be a correct answer. Okay, that's why we do not class it as a cell. The next one we can write they do not have cytoplasm, or we can write they do not have nucleus, or we can write they do not have a mitochondria like most eukaryotic cells, or we can write they do not have ribosomes. A vaccine can protect humans from a viral disease. What does the vaccine contain? A toxic form of a virus. A vaccine never contains a toxic form of virus because otherwise it will, damage the host. So, it contains a weakened form of the virus. Exactly correct. If we give the weakened form of the virus will not be able to take in effect of that original virus. However, the body will be able to detect its antigens, which is the foreign protein of the virus, and the body will be able to produce antibodies against those antigens and thereby protect itself from future invasion. Neither do we, inject an active form of virus, because this will then lead to that particular viral infection. We want to get rid of the virus, alright, by building up immunity. That's why we do not give active form of the virus. In some cases, a first vaccination needs to be followed by a second vaccination sometime later, which graph shows how the concentration of antibodies in a person's blood changes after the first and second vaccination. In the first vaccination, the level of antibody will increase after a certain amount of time. However, then it will start to decrease again. And then during the second vaccination, because there are memory cells present, the level of antibody will then rapidly rise during the second vaccination, because there were memory cells present which took responses much faster than the first vaccination, and the concentration of antibody produced will be much higher. The second and the third graph are wrong, because in the second graph we can see that as the time progresses with the vaccination and the second time, it is a linear, alright, straightforward. With time the concentration of antibody increases. However, which is not true, because after the vaccination is done, that antigen will no longer be present in the blood, so there is no need for increasing antibody concentration indefinitely. Next, antibody concentration in the beginning of vaccination is very high, which is nearly impossible, because the person was not infected with that particular virus before. So how would the person have antibody for that particular virus? That's why this is wrong. Tobacco mosaic virus TMV causes disease in plants. TMV affects the rate of photosynthesis in plants. Which part of a plant shows discoloration caused by TMV? Now, since there is a hint in this particular question, we can see that this is a disease in plants and it decreases the rate of photosynthesis. Photosynthesis is done by chlorophyll or chloroplast. So if the chloroplast or the chlorophyll is damaged, then obviously the leaf is damaged, because leaf contains contains chlorophyll or the chloroplast. Flower or the root does not contain chloroplast for photosynthesis. Table 1 shows the rate of photosynthesis in 4 different tobacco plants. We can see tobacco plant A, the level of TMV infection in plant is none. There is none present there. Rate of photosynthesis is the highest. Whereas if the level of TMV infection is high, the rate of photosynthesis decreases to arbitrary units of 3. So we can see as the infection increases, rate of photosynthesis decreases. You should label the y-axis, add the correct scale to the y-axis, plot the data on table 1 and label each bar. So one of the bar is done for us, which is the none. So the other ones will be B, C, D. In the mild case, we can see 13. Okay, so we can see that in the x-axis there is no labeling. So let's put the labeling on the x-axis. Sorry, the y-axis there is no labeling. So let's put the labeling on the y-axis. The y-axis consists of rate of photosynthesis in arbitrary units. Since the first none is up until 15, so let's label it 15, and we can see there is a total of 6 boxes for 15. So we will have to divide the 15 with 6 and the answer will be 2.5. That means each box will represent 2.5. So in the case of mild infection, 13 can be represented . What conclusion can be made from the data in Table 1? So from the data in Table 1, we can say as the level of infection with TMV increases, the rate of photosynthesis decreases. Explain why a high level of TMV infection reduces growth in plants. A high level of TMV infection reduces the amount of chlorophyll within the plant. Since the chlorophyll decreases, there is less glucose produced and less proteins and starch is made within the plant. A cactus is a plant that lives in a dry environment. Figure 6 shows a cactus plant. We can see figure 6. Give one adaptation shown in figure 6 that helps to prevent the cactus from being eaten by animals. We can see the cactus plant have a lot of spikes or thorns, which will hurt the herbivorous animal when it will try to eat it. So the answer will be it has spikes, or we can say thorns or prickles. A plant may produce poisons that make animals unwell. What is the type of defense mechanism? Poisons are considered chemical because they can chemically activate a particular process within the body. So they will be a chemical defense mechanism. A mechanical form of mechanism of defense would be thorns and spikes, which will cause physical harm, and the mechanical will lead to physical harm. So both of these other options are incorrect. Some desert plants only grow leaves after it has rained. As soon as the soil dries out, the leaves fall off. How could the leaves falling off the plant be an advantage to a plant that lives in a dry environment? Leaves contain a lot of stomata, and thereby it will lose a lot of water. So let's see the points. The plant is less likely to reproduce. Well, the leaves are not a reproductive organ, so we can just cancel it. The plant will not lose as much water. That is correct. Leaves provide a very large surface area. So when the leaves fall off, the large surface area goes away and the plant will not easily dry. The plant will photosynthesize faster. No, however, the plant will suffer in terms of photosynthesis. Okay, but in living in a dry condition, photosynthesis is not the first goal of the plant. The first goal should be survival. So losing the leaf loses the surface area where the water evaporates and increases the chance of survival. The stem of a cactus is green. What causes the green color in the stem? Obviously, the stem contains a lot of chlorophyll. Since it contains a lot of chlorophyll, the chlorophyll or the chloroplast that contains the chlorophyll gives the stem color green. What is the advantage to the cactus of having a green stem? Having a green stem, means that the stem is able to photosynthesize and make some sugar or glucose molecule. The stem of a cactus contains many different tissues. What name is given to the group of tissues working together? A group of tissues working together is going to be called an organ because different groups of tissues are working together. If why organism and organ systems are not correct, a group of organs working together can form an organ system, so that's why it's incorrect, and a group of organ system working together can form organism. So these two are a higher level of organization compared to the organ. Name one substance transported through the xylem in the stem of the cactus. Xylem is going to carry water and mineral ions. So both will be correct answer. You just need to write any one of them. Name the tissue that transports dissolved sugar through the stem of the cactus. Now, the transporting of dissolved sugar is the job of a phloem. Carbohydrates are needed as a part of a balanced diet. Which formula shows glucose? For students, glucose, the formula is very much necessary. Just remember, glucose consists of 6 carbon joined with hydrates of that carbon. Hydrate means water. So if there are 6 carbon, there will be 6 water, which means there will be 12 hydrogens and 6 oxygen. As you can see, the other options are obviously wrong. For example, CO2 is carbon dioxide and H2 is water and O2 is just oxygen. So these 3 are obviously wrong, so you can always choose the correct answer very easily. Which type of enzyme breaks down starch? Now, the enzyme that breaks down starch, we have to understand that a starch is a carbohydrate. So since the starch is a carbohydrate, that means it will be broken down by the type of enzyme carbohydrase. Now, what are the other responses? The other response here includes lipase. Now, we need to know lipase breaks down lipid and protease. Protease breaks down protein. A student investigated the effect of temperature on the activity of the enzyme amylase. Figure 7 shows the apparatus used. We can see test tube. We can see a beaker Q which contains water at 10 degrees Celsius. So beaker Q is acting as a water bath. We can see 5 cm³ of the starch solution and 1 cm³ of amylase solution. Both are incubated at the same temperature. The method that is used: set up the apparatus as shown in figure 7. After 5 minutes, pour the starch solution into the amylase solution and mix. So we are putting them for 5 minutes in the same temperature so that they all acclimatize or come to the same temperature before we start the experiment. Remove 1 drop of the amylase starch solution mixture and place it onto a spotting tile. Immediately add 2 drops of iodine solution to the amylase starch solution mixture on the spotting tile. Record the color of the iodine solution added to the amylase starch solution mixture. Repeat steps 3 to 5 every minute until the iodine solution is yellow brown. So technically what we are doing is we are performing a iodine starch test every minute. Now if the solution turns blue black, that means there are still starch present and the amylase did not finish its job. When the solution turns yellow brown, that means amylase has break down all the starch and the solution then becomes yellow brown because iodine has a yellow brown color. Now name the apparatus Q in figure 7. So we can already see Q is a beaker, so we will just name it beaker. Now because the Q is used as a water bath, so water bath is also a correct answer. Why were the starch solution and the amylase solution left for 5 minutes before mixing them together? So that both solutions could reach 10 degrees Celsius. Exactly. We want both of the solution at the same temperature. This is correct. Now let's look at the other responses. So that the student could calculate a mean. No, we are not trying to calculate a mean of the temperature. Alright. So this is a wrong statement. So that the student could repeat the investigation. Well, the main purpose is for both of the solution to reach the same temperature. So point number 1 is correct, and this third point would be repeating investigation. We can still repeat it, however that is not a correct statement. So that the student had time to draw a table of results. Well, the student can draw the table of result before the experiment. Anyways, so the first option is the correct one. Figure 8 shows the result. We can see a spotting tile and we can see it starts from 0 minutes and then 1 minute, meaning this direction is the one that we have to follow. Alright, then we can see iodine starch solution and amylase starch solution mixture. Alright, this lighter one indicates yellow brown and the darker one indicates blue black. So we can see after 1 2 3 4 5 6 7 8 9 minutes, we can see that the reaction has finished. How many minutes did it take until the iodine solution and amylase starch solution mixture was yellow brown? Because we are asking the question how many did it take for it to become yellow brown, so it will be 10 minutes. How could a more accurate time be obtained? Add more iodine solution to the spotting tile. Test the mixture with iodine solution every 30 seconds. Test the mixture with iodine solution for more time. Use 2 drops of amylase starch solution mixture in each test. Now using 2 drops has no added effect. To test the mixture with iodine solution for more time will not have any added effect because the reaction has finished after 10th minute. Okay, and the first point, add more iodine solution to the spotting tile, will have no effect because it will still give the same blue black or yellow brown result. So testing the mixture at a closer time interval, meaning that we could tell whether the reaction has finished at 9 and then 30 seconds. So 9 minutes and then 30 seconds. So we could tell that difference. So that gives a higher resolution of the result. So testing the mixture with iodine solution every 30 seconds would be the correct answer. The student repeated the investigation at 5 different temperatures. Table 2 shows the result. We can see temperature from 20 to 80 degrees Celsius. Time taken for the iodine solution mixture until the mixture was yellow brown in minutes was ranging from 5, it decreased to 2 and then increased to 12, and at 80 degrees Celsius there was no the entire solution remained black even after like 12 minutes. Okay, which temperature did the enzyme work quickest at? Now definitely the temperature at which we could see the yellow brown the fastest is 35 degrees Celsius. So that will be the correct answer. Explain why the iodine solution remained blue black in the investigation at 80 degrees Celsius. Now an easy explanation for this would be at a very high temperature like 80 degrees Celsius, the enzyme amylase is denatured, so the starch is not at all broken down by the enzyme. A high cholesterol concentration in the blood can lead to blockages inside arteries. The coronary arteries supply blood to the heart muscle. Figure 9 shows the coronary artery with a blockage. We can see the direction of the blood flow and we can see the blockage that is narrowing the blood vessel. Why could the blockage in figure 9 cause the cells in the heart to die? The blockage restricts the flow of blood, so less blood flow through into the heart muscles and tissues. And since less blood flows, less oxygen reaches the heart muscles and the heart keeps on working, so it slowly produces more and more lactic acid, which then basically kills it. However, we only need to talk about that less blood flows to the heart muscles and less oxygen reaches the heart muscle tissue. Doctors can measure the concentration of cholesterol in the blood. Table 3 shows 4 different blood cholesterol categories. We can see blood cholesterol concentration in millimole per dm cube. Less than 4.6, cholesterol category is low. And then we have 4.6 to 5.0, which is normal, and 6.2 and above, which is high. Okay. Figure 10 shows the blood cholesterol concentration of 4 people. Okay. We can see person A, B, C, and D. A has a blood cholesterol concentration of 4, B has 6.4, and C has 4.7, and D has 5.6. Which person is in the medium cholesterol category? So medium cholesterol category is categorized when someone has between 5.1 and 6.1. So 5.1 and 6.1 would be person D. Which person is most at risk of having a heart attack? The person who has the highest cholesterol level, alright, would be at most risk. So cholesterol category 6.2 and above would be at a high risk of heart attack. So person D has more than that. Person B has more than that. So person B will be at a higher risk of heart attack. Give a reason for your answer to question number 5.3. Person B is more likely to have blockages because the person has very high cholesterol level. So the person B's blockage could be the biggest. The blood cholesterol concentration of person D is greater than the blood cholesterol concentration of person A. Calculate how many times greater. So person D has 5.6, whereas person A has 4. So we will have to do 5.6 divided by 4, which will give us 1.4. So number of times greater would be 1.4. Figure 11 shows a stent can be used to treat a person with a blockage in coronary artery. Direction of blood flow, and we can see the stent is opening up the blockage and making the blood flow normal in the coronary artery. Explain how a stent works as a treatment for a person with a blockage in coronary artery. The stent, as we can see in the figure, is widening up the artery so more blood can flow through the artery without any restriction. So more oxygenated blood will be able to reach the heart muscle tissue, and as a result more oxygen will be supplied to the heart muscle tissue as well. Guys, only the points the stent widens the artery and more blood can flow through the artery will give you the marking point. Patients are given anti-clotting drugs after they have a stent fitted. The drugs help to prevent clots forming in the blood. Which part of the blood starts the blood clotting process? Now, blood clotting is started by platelets. The other responses are antibodies. We know antibodies are needed to fight, foreign pathogens basically. So plasma is the liquid part of the blood, and then red blood cell is necessary for carrying oxygen. However, platelets are necessary for blood clotting process. When a stent is fitted, the doctor gives the patient an injection of anti-clotting drugs. The patient then takes an anti-clotting tablet every day. Anti-clotting drugs are very effective, can take a week to begin working fully, have been used for over 60 years, cost very little to make, do not work effectively if the patient eats certain types of food. The patient must have their blood tested every few weeks to check that the anti-clotting drugs are working. Evaluate the use of anti-clotting drugs in patients who have had a stent fitted in them. So there are advantages for taking the anti-clotting medication. One of the anti-clotting medication advantage will be the person needs to take only one medicine per day, one tablet per day, and only a tablet is so easy to take, alright, and it's not painful to take at all. The drug is effective, so the person is less likely to get a blood clot. The drug is cheap, so it is less cost for the national healthcare system. The drug has been used for a long time and it is more trusted by people. However, the disadvantage also lies there. The patient have to make sure they always have a supply of the drugs, because if the level of the drugs falls, then the patient will suffer. The patient could forget to take drugs every day. Patient could still get a blood clot in the first week because it takes at least 1 week to take effect. There are restrictions on patient's lifestyle because the patient cannot eat certain types of food. The patients may get a blood clot if they eat the wrong food, and there are risks associated with puncturing of skin, because then there can be more blood loss when the skin punctures. Alright, the patient may have a fear of needle when the patient is given an injection, and there is a higher risk of bleeding and bruising if the patient gets like into any sport events or normal day-to-day activity. All right, there will be a higher risk of bleeding and bruising. All of these are correct point. However, to get the 4 marks, we'll have to write 2 points for disadvantages and 2 points for advantages. However, I'm going to mention you the points. You can choose which 2 points you want to write. Figure 12 shows a model used to demonstrate human breathing. We can see a bung in the glass tube, and then the glass tube is connected to 2 balloons, a glass bell jar, and a rubber sheet. Which part of the breathing system is represented by the glass tube? The glass tube is representing like the trachea because it is bringing the air like the trachea into the lungs. The balloons are acting like the lung, and the alveoli and capillaries are actually not represented in this diagram in any form. The model in figure 12 represents the human breathing system. The teacher said the model does not represent the human breathing system very well. Give 2 reasons why the teacher is correct. The teacher is correct mainly because there is only 1 air space per balloon, so alveoli is not represented. Blood vessels or capillaries are not represented as well. Bronchioles are not represented in the diagram. The glass tubes are not flexible like trachea. The bell jar does not move during the breathing like the ribs. The ribs have gaps between them which the bell jar doesn't have. The rib cage contains muscles which the bell jar doesn't have, and the pleural cavity not represented in this particular diagram. So guys, only 2 points will be needed to score this mark. However, I'm going to give you all of the points. You can choose any one that you want to write. A scientist investigated the effect of exercise on breathing rate. This is the method used by the scientist. Record the breathing rate of 10 male non-smoker at rest. Tell each man to run on a treadmill at the same speed for 8 minutes. Record the breathing rate for each man every 2 minutes. Continue to record the breathing rate for each man for 4 minutes after he stops running. Give 2 variables the scientist controlled in this investigation. The scientist has specifically chosen a particular exercise, which is, the treadmill, and the speed at which each man are running on the treadmill. And also the scientist has chosen the type of exercise which is using the treadmill. The scientist has also taken males, gender, sex into consideration. Then the scientist choose all non-smokers. So that is also something the scientist is using as a controlled variable. And also every man were allowed to run for 8 minutes, so which is also a controlled variable. Figure 13 shows the data collected from one of the men. Breathing rate in breaths per minute and time in minutes. We can see as the time in minutes increases, breathing rate in breaths per minute increases up until 8 minutes and then decreases up until 12 minutes. Calculate the percentage increase in men's breathing rate between 0 minute and 8 minutes. So to do this calculation, we have to do percentage increase breathing rate at 8 minutes minus breathing rate at 0 minute divided by breathing rate at 0 minute. So breathing rate at 0 minute was already 20 BPM or breaths per minute, and breathing rate at 8 minute is, we can see it's 42 BPM breaths per minute. So it will be 42 minus 20 divided by 20 into 100. So the percentage increase after calculation we find out is 110%. It is important that we extrapolate this particular data. And show it because the question contains 3 marks. It will be important for us to get that particular mark. So at 0 minute 20 BPM, at 8 minutes 42 BPM. So the percentage increase is 110%. Explain why the man's breathing rate increased when he was running. The man's breathing rate increased when he was running to get more oxygen into the blood. The oxygen will be used for respiration, for releasing energy during muscle contraction. We can also say a different category of answer. The increase in breathing was to remove the carbon dioxide dioxide which was produced during the respiration. Give one measurement that could be taken to show a different effect of exercise on the body. Do not refer to breathing rate in your answer. Since in this particular answer we cannot talk about the breathing rate, so we can talk about the heart's pulse rate, the number of beats per minute for the heart. We can also talk about the volume or the depth of breathing during the exercise, and the volume of sweat that is produced during the exercise, or the body temperature increase during the exercise. The men in the investigation were all non-smokers. Give one effect that smoking can have on the body. Smoking can cause lung cancer, or we can say smoking can increase blood pressure. Smoking can lead to lung diseases. Smoking in pregnant women leads to low birth weight in babies. Smoking increases the risk of cardiovascular diseases. All of these are possible answers, and you can write any one of them. A student prepared some animal cells to view using a microscope. Figure 14 shows the student preparing the cells. We can see the figure 14 and how the cells are prepared. Name two pieces of laboratory equipment the student could have used to prepare cells to view using a microscope. We can take this particular information from the diagram, which is a microscopic slide that is used here, and a cover slip. The other answers are dye, so that we can color that particular piece of we can color that particular piece of cells, a mounting needle, a dropper, all right, to put in solution. Sometimes we can put in saline solutions, or the dye, the solution of the dye, a scalpel to cut the thin section of that particular piece to be viewed, and forceps. All of these are correct answers, so I'm just going to mention them. Answer any two of these particular points. Figure 15 shows the student's light microscope. We can see the part A, we can see the part B. Name of the part A is asked in this question. So the part A has a lens which we use to view that particular, microscopic, or whatever is in the microscopic slide. So it's called eyepiece lens. And the bottom one is called objective lens. And part B is actually the adjustment to focus. The function of the part B is to focus the image. The student tried to look at the cells using the microscope. Suggest one reason why students could not see any cells when looking through part A. It could be due to many reasons. One of the reasons could be there were no cells in the field of view, or we can say the slide was not in the correct position, or we can say the mirror was not in the correct position, or the objective lens did not click into its place, or we can say the student is looking at a large air bubble, which is why sometimes you won't be able to see the image, or we can say the microscope is not focused. Any one of this particular point is correct. Red blood cells are specialized animal cells. Compare the structure of a red blood cell with the structure of a plant cell. Since this particular question is about comparison, so we can say the similarities and differences between the red blood cell and a plant cell. The way we can start is we can start with the similarities. We can say both red blood cell and plant cells have cytoplasm, cell membrane, and pigments, although the pigments are different. And then in terms of their differences, we can then go on that red blood cells have no nucleus, whereas plant cells have nucleus. We can say red blood cells have no cell wall, plant cells have. Plant cell has cell wall. Red blood cell is a biconcave disc, and there are many different shapes of plant cell. Red blood cell contains hemoglobin, whereas plant cells do not contain hemoglobin. Red blood cells do not contain chlorophyll, but plant cells may contain, some of them, most of them actually, may contain chlorophyll. Red blood cells has no chloroplast. Plant cells has chloroplast. Red blood cell has no permanent vacuole. Plant cell has permanent vacuole. And red blood cells are much smaller than the plant cells. So this could be the indicative point for the differences. I'm going to give you all of the points. However, from similarities write 3, and from differences at least write 4 to make total of 7 points so that the full marks can be achieved. When placed into a beaker of water, a red blood cell bursts. A plant cell does not burst. Explain why the red blood cell bursts but the plant cell does not burst. When water enters the cell by osmosis, the plant cell has cell wall which prevents it from bursting. However, the red blood cell does not have any cell wall, so it swells and it bursts. A student investigated the effectiveness of 3 different antibiotics. Figure 16 shows how the student set up an agar plate. We can see bacteria growing on agar, and we have a filter paper disc containing antibiotic A, filter paper disc containing antibiotic B and C in a petri dish. The student used aseptic techniques to make sure that only one type of bacterium was growing on the agar. Describe 2 aseptic technique the student should have used. Guys, when we are asked about aseptic techniques, we mainly need to think about sterilization. So we must use sterilized equipment, alright, and sterilize the surfaces before we use it. We need to use sterilized agar that does not contain any bacteria from before, alright. So we need to heat it to higher temperature to kill all of the bacteria. We need to secure a lid of the petri dish with adhesive tape so that outside bacteria are unable to enter. We need to only lift the lid of the petri dish a little when setting up the plate so that we do not introduce outside bacteria. Or what we can do is we can lift the lid of petri dish at an angle when setting up the plate so that we can minimize the introduction of bacteria from outside. We just need to write 2 of these points. However, I'm going to give you all of them. The student placed the agar plate in an incubator at 25 degrees Celsius for 48 hours. Figure 17 shows the agar plate after 48 hours. We can see the area where the bacteria are not growing. When we look into the picture, we see C has the greatest area and B has the minimum amount of area. Which antibiotic is the least effective? Give a reason for your answer. So we would say the least effective one is B, and the reason for that is going to be it kills the fewest amount of bacteria, or we can say B has the smallest area where no bacteria were growing. Calculate the area where no bacteria were growing for antibiotic C. We need to use pi is equals to 3.14. To do this calculation, first of all, we need to calculate the radius of the antibiotic C. So to calculate the radius of antibiotic C, we are going to, from the center, we are going to measure up until the circumference. And in this case, the value is 1.1 centimeter. So once we get 1.1 centimeter, so we're going to write down R is equals to 1.1 centimeter. Then because we have to do the calculation, and the calculation is best done with either cm or millimeter. All right, so let's say if we decide to do it with cm, then we need to then use the equation. The equation is for for the area for a particular circle will be pi r square. Once we use the pi r square, then we have to use the pi value of 3.14 into r square, which is 1.1 whole square, and that will give us the calculation. This is equals to 3.7994 cm square. Okay, now for our exam, all right, we have to also make sure that our answer is up to 3 significant figures. So since our answer, all right, is 3 significant figures, so we will do this is equivalent to 3.79, so we can do it 3.80. Okay, all right, then the area will be 3.80. Guys, exam significant figure is 3 significant figure. However, in biological calculations where 3 significant figure does not apply, we can keep the value to be 3.799, and we can mention that as well. And if the unit and the calculation is correct, it will score 5 marks. Suggest one way the student could improve the investigation. The student can repeat the entire calculation process and calculate an average. Now the student either can do this, or the student can use a control disk in the second repeat of the experiment, or the student can repeat the same experiment with a different type of bacteria. All right, this is how the student can show repeatable result with different bacteria and the antibiotics. So all of these will be accepted answers, so I'm going to give you all of them. Body mass index or BMI is a way of finding out if a person's body mass falls within a healthy range for their height. Table 4 shows information about 2 people. You have a person A, which has a body mass in kilogram of 63, height in meter of 1.65, BMI in kilogram per meter square of 23.1. For person B, body mass of 692 and height in meter of 1.71 and BMI in kilogram per meter square of X. So body mass in kilogram on the x-axis and height in meters in the y-axis is represented. Okay, now the question says which is the BMI category of person A in table 4. So person A has a mass of 92. So what we're going to do is we're going to find out 92 from the diagram, which approximately this is 95. So this could be somewhere in the middle. So this is 92. Then we are going to go, all right, straight forward up until the height. Now the height is 1.71. So 1.72 and 1.70, this is where it lies. So we would have to travel just where both of these particular lines will meet. Okay, now both of this particular line meets at a particular point where it says that the person is obese. So we will say the person is obese. Calculate the value of x in table 4. Okay, so x in table 4, the calculation is 1.71 and 92. So what we have to do is, first of all, we will take the BMI equals to the body mass is in this case 92 divided by height, which is 1.71 whole square. So once we do that, our answer comes 31.46. all right, which rounded to 3 significant figure will be 31.5. The scientist thinks there is a link between BMI and life expectancy. Table 5 shows information about predicted life expectancy of men after the age of 50. For example, the BMI category, which is normal, overweight, obese, and clinically obese, we have a predicted number of years of living in good health after the age of 50, ranging from 19.06 with clinically obese 13.07, and predicted number of years living in bad health after the age of 50, with normal it's 4.98, which increases to clinically obese to 10.10. Describe two patterns shown in table 5 about the effect of BMI category. So we can say the two patterns is the higher the BMI. If we have a higher BMI, then we have a lower life expectancy with good health. And if we have a lower BMI, if we have a higher BMI, we have a higher life expectancy with bad health. So we can write the answer. There are 3 possible points that we can give this answer. So I'll give you all of the points. The higher the BMI category, the lower the number of years living in good health. The higher the BMI category, the higher the number of years living in bad health. The higher the BMI category, the lower the life expectancy. The number of people who are obese in the UK is increasing. Explain the financial impact on the UK economy of an increasing number of people who are obese. As the number of obese people increases within a particular population, such as the UK, alright, the cost for national healthcare system increases because the governments need to pay more money for additional surgery and medication and hospital stay to treat stroke or complications like diabetes. Or we can say a patient needs to take more time off work if the patient is unwell, and the employer or the government have to give that financial support to that particular employee. A person who is obese is more at risk of arthritis. Arthritis is a condition that damages joints. Suggest how arthritis could affect a person's lifestyle. Arthritis can lead to a lot of complications. One of the major complications is movement issues. So the person will have movement issues. The other possible answer will be for this particular point, because it is a lifestyle problem, alright, because it leads to the problem in the person's lifestyle. They are loss of job or income. The person may have permanent disability. The person may have mental health impact, alright, due to the lack of movement. The person need to visit doctor and take medication more often, or the person may need to undergo surgery to to improve on that particular joint that is impacted by arthritis. So all of these are possible answers. Just choose any one that suits you best and write it in your exam. A person who eats a diet high in saturated fat might become obese. Name 2 health conditions that might develop if a person eats a diet high in saturated fat. Do not refer to arthritis in your answer because we have already done it. Okay, great. So we can say the person can develop type 2 diabetes, because obesity leads to type 2 diabetes. Definitely the person may develop cardiovascular disease, or the person may have a heart attack or stroke. So these are all of the possible answers. Guys, that's all for this particular question paper. Thank you for joining the video. See you in the next video, guys. Bye-bye.